The last video ended here, when comparing prokaryotes and eukaryotes in the process of transcription. Hopefully at this point you have already watched this video link down here, and you've already filled in your table with the key differences between eukaryotes and prokaryotes when it comes to transcription. So what happens next now that we have mRNA? Kind of a note, a side note, is that I just showed you one DNA molecule and one RNA polymerase on it. But in reality, or in actuality, that multiple RNA polymerases can be working at the same exact time on a single gene. And that's what you see in this picture. So with my mouse, I'm following a strand of DNA. All of those black blobs that you see are RNA polymerases. Now, each complex is working just like we talked about: initiation, elongation, and termination. But it's just happening multiple times. So you're getting many, many copies of this mRNA having been transcribed from DNA. And this happens in both prokaryotes and eukaryotes. Now, this mRNA isn't really ready to go in eukaryotes. In prokaryotes and in bacteria, it is. That mRNA is transcribed from DNA, and that mRNA is ready to go to be translated into a protein. However, eukaryotes have some processing that has to happen. That mRNA, as soon as it's released from RNA polymerase, is not ready to go. It cannot go to the ribosome yet in order to be translated. It has to go through some steps called RNA processing. So in eukaryotes, we really kind of refer to this type of RNA as pre-mRNA because it doesn't really work yet, which is why we kind of put that prefix in front of it. This image is kind of showing you an idea of, here is what this pre-mRNA looks like. We have DNA and then this pre-mRNA, and then you add some things to the front, things from the side, take some things from the middle. So quite a few modifications that this pre-mRNA has to go through before it's ready to translate into proteins. So at the bottom, this is kind of what we're trying to get to. This is what we're trying to get our mRNA to look like. So there's two features that are showed on this slide. The first one is the 5' cap. As the name suggests, it's something that's going to go on the 5' end of this pre-mRNA. What this cap is, it's guanine, but it's been methylated, as in we've added a methyl group. Methyl is CH3. But it's a methyl group. There's also 3 additional phosphates, and you see that down here. Here's the G for guanine. This M is representing methyl, and then our 3 phosphate groups. The reason mRNA needs it is actually for a couple of reasons. One, it's going to help protect this pre-mRNA from ribonucleases. Ribonucleases are enzymes that break down RNA. And it's actually kind of a defense mechanism for our cell. If we see mRNA or any kind of RNA floating around, that could have potentially been a foreign invader. So ribonucleases, their job is to break it down. But by putting on this methylated cap, this 5' cap, it kind of tells the ribonucleases, hey, I'm supposed to be here. Yes, I'm a loose strand of RNA, but I actually have a function. The other way it serves, that we'll talk about later in translation, is it kind of serves as an attach here sign. This is what's going to attach to the ribosome to start translation. So that's one side of our pre-mRNA, trying to get it ready to be full-blown mRNA. On the other end of this molecule, what you notice is there's a 3' poly-A tail. And by poly-A, we mean many A's or many adenines. And by many, I'm talking about hundreds of adenines being added all along this very long tail. The enzyme that catalyzes this reaction is a poly-A polymerase. Polymerase meaning you're making a polymer. So a polymer of many, 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 many A's. Essentially what it is a kind of almost like a telomere. If there's any nucleases around trying to cut the DNA or the RNA, well, it has all this junk RNA at the end that it can cut off. Also, having all these adenines in a row will actually help it get out of the nucleus. It's a signal that those nuclear pores need. And it also helps in translation, which we'll talk about in a little bit. Now remember, in eukaryotes, I mentioned in the termination phase that this mRNA will have a poly A region that is not the same as this poly A tail. That poly A region would be along here. So somewhere in this untranslated region. You have that initial poly A that's much, much shorter than this tail. And that's what encourages an enzyme to approach the RNA polymerase to cut that mRNA away. So the poly A that was used in termination is different than the poly A you see here in this tail. Now again, prokaryotes do not undergo this. They do not need these special features. The second thing that eukaryotes undergo, so adding the cap and adding the tail is one thing. The second thing eukaryotes have to do is we actually have extra DNA that doesn't do anything. Well, I shouldn't say doesn't do anything, but our proteins won't need that section of DNA. We'll kind of talk about their function in a little bit. So mRNA is our copy of our DNA, and there's two components of the mRNA. We have exons and we have introns. So just looking at this diagram down here, it's showing our DNA and our RNA. Here's an exon, here's an intron, here's an exon, here's an intron. They're kind of alternated. There's little bits of each. Those exons carry the information we need. The exons are the raw information we need to make amino acids. Introns, however, are just extra. They don't really code for that protein at all. And we got to kind of remove them, because if we took it to the ribosome like this, the ribosome would be adding all of these extra amino acids that aren't supposed to be in that protein and would change the shape of that protein. So a way to remember which one is which one is the good one or the coding one and which one isn't, I think of exons as expressed. You know, this is the part of that RNA that's going to be expressed, that we're going to get amino acids out of. However, the introns are intervening. They're in the way. So it's kind of a way to help memorize or remember which one is which. So how do we get rid of these introns? We don't need them, but how do you get rid of them? So of course, we have enzymes. We have tons of enzymes in our body. We have enzymes that are specifically designed to take care of this problem, to get rid of these introns. So on our introns at the end and at the beginning, there's just a quick little code that indicates to us or indicates to our cells that they are introns. And you can see that here in this picture. So here this is kind of showing, oh, this is that key sequence. So there's one here, and there's one almost near the end. So short little sequences that are going to tell our cell, all right, here's the end of my intron. The next thing that gets added are small nuclear ribonucleoproteins, or snRNPs. And I do recommend kind of making a vocabulary sheet, because we do talk about so many different words in here and so many different terms and complexes. So snRNPs consist of two different things. snRNA, which is small nuclear ribonucleic acid, and then proteins. So that's how you kind of get this snRNPs, that snRNA plus proteins. And their whole job is just to recognize that short sequence. And that's what you see in this image. That here's are these snRNPs that are attaching around this intron, getting ready to get rid of all of that extra material that we don't really need. And then what you see in this bottom image is that all of these snRNPs are coming together. They're kind of bonding to each other, and what you notice is it's kind of looping off this intron. We call this new complex of proteins, we call that the spliceosome. So splice, when you splice something, you're cutting something out. And that's exactly what the spliceosome is going to do. It's going to cut part of that intron out, or really the entirety of that intron. So the spliceosome, its whole goal is just to cut away all of that intron. And that's what you see right here. That spliceosome has cut off one side of that intron, and it's going to cut off this other side of the intron. Now this spliceosome, again, is doing this cutting, but specifically, I said it had this snRNA, a nucleic acid, and it has proteins. Now we've talked so much about proteins catalyzing reactions, and proteins do this, and proteins do that, and proteins are amazing. But it's really cool because in the spliceosome, it's actually the snRNA. It's an RNA molecule that does the cutting. And we've used this term before, but it was many chapters ago. This snRNA is a ribozyme. So an enzyme was a protein with catalytic ability. Well, a ribozyme is an RNA with a catalytic ability. We talked about this when we talked about the RNA world hypothesis, that everything came from RNA. RNA has a template, so it can make copies like we saw in transcription, but it can also catalyze reactions. So it's actually the small nuclear RNA that's in this spliceosome that is cutting away that intron. And you see it makes this loop, and then it gets cut out, taken away, and then now you have this beautiful mRNA that only has these exons. But why have that anyway? Like, what's the point of having these introns, these in-the-way materials? Is there any benefit? Now, this is actually a current area of research. There's no definitive, it does this, does this, does this. We have a couple hypotheses, some of which have been supported, but it's still kind of guesswork, especially on how did they even get into our DNA to begin with. So one thing we have noticed about our introns and our exons is that it's not always the same. When we do this mRNA processing, the introns that are labeled right here, you're like, oh, well, we get rid of those introns. Yes, but sometimes the spliceosome doesn't get rid of that intron, it gets rid of another intron, or it'll get rid of an exon. So looking at this example here is what our gene, our DNA looks like: an exon, intron, exon, intron, exon. And it goes through transcription, it goes through RNA processing, and here's the final result, just exon 1 and 2. Our spliceosome actually got rid of both introns and exon 3. And so these two coding regions are going to make one type of protein. Well, if me as a spliceosome, I could get rid of everything here in the middle, and so now I have exon 1 and exon 3. Well, that's going to be a completely different protein. So maybe the reason we have these various exons and introns is that really our cells can kind of pick and choose these different pieces to make different proteins. So I can make many, many proteins from one single gene just by putting together different blocks. Another hypothesis as to what is happening is that maybe each exon, each coded piece of gene, is actually a different domain or a different, specific section of a protein. So looking down here, we have 3 different colors representing the 3 different exons, and there are 3 distinct areas of this protein. Maybe each one just kind of has a very specific function. Maybe one of these serves as an active site, or an allosteric site, or some other bonding site. So maybe it's the cell's way of kind of keeping everything separated and organized. So just as an overview of eukaryotes and RNA splicing, here we have a micrograph, so an actual scientific microscopic image of DNA. And you can actually see these loops, these extra bits of information that aren't needed. And here in this digitized version, the red was that original RNA, that pre-mRNA. And yellow is going to be what's completed once you get rid of all of those loops. So we're going to stop here for now, review as much as you need to before starting the next video.